Good day. You can see these are the test cross notes, and some students find these to be a little complicated, so pay close attention. So test cross is used to determine the genotype of an organism with a dominant phenotype. So that means something we can see that's a dominant trait. The issue is, if it was a recessive phenotype, then we know we just have recessive genes that combine to be homozygous recessive. But really what we're trying to do is figure out, is it homozygous or heterozygous? So this typically would not be done in humans. It would be done if you're breeding plants or certain animals. Well, the process is to mate the organism, and this is the one with the dominant phenotype, with someone that has a recessive trait, and then you examine the offspring, and this would be done multiple times. So what we're going to show you is, using Punnett squares, what are the possible results, and what does that mean? What do these things tell us? So we're going to go simple with pea plants tall and short. So we want to determine, is it homozygous dominant, capital T, or heterozygous, capital T, small t. So that means we would cross the tall pea plant with a short pea plant. Well, there's two possible outcomes, well, two possible crosses, I should say. So if we had dominant and it was homozygous, big T, we can see only thing we can get from the cross is the tall trait. They'd all be heterozygous and all the pea plants would be tall. But if we crossed with the recessive trait and we had a heterozygous, we would see half are tall and half are short. So what really matters is both of these crosses give the dominant trait, the tall trait, but only if it's heterozygous do we get the recessive trait, the short trait, appearing, and that becomes very important. So that's basically what I've just said. If the unknown genotype is homozygous TT, we just simply have no chance of getting short. It's going to have the dominant trait. Here is a 50-50 chance of dominant and recessive. Well, this turns out to be very important. If all the offspring are tall, meaning, hey, we got tall or tall, we do not know for sure which genotype the parent has. Were they homozygous or heterozygous? But if we do the cross many times, we would get more confidence that they're homozygous, because the other way, it's quite likely we would have got the recessive trait, but we never know for sure. So if we did this 5 times and always got dominant, yeah, it's probably homozygous, don't know for sure, wouldn't be totally confident. If we did this 100 times and only got dominant trait, then we'd be pretty confident, a lot more confident that it's homozygous. But for that, we have to do it many times. On the other hand, as soon as the short trait appears in the offspring, we know for sure it was heterozygous, because the homozygous dominant will not allow us to get that recessive trait, the short trait, appearing. So it only took one to convince us that it was heterozygous for the unknown genotype. So you will be asked questions about that. I hope it makes sense.